Hi, my name is Emily. Today I'll be talking to you about my project, Deodorization of Microalgae for Staple Crop Adaptation. Nearly all agriculture is terrestrial. Over 98% of global caloric production comes from land based crops. This hyperconcentration has two consequences: food security and sustainability. Food production shocks are increasing in frequency, driven largely by extreme weather, and meeting demand from land alone drives soil degradation, deforestation, and biodiversity loss. Marine agriculture solves crop hyperconcentration. Right now, marine food production is oriented around fisheries. Marine phototrophs remain critically underexplored. In particular, microalgae is competitive in both nutritional profile and production efficiency, positioning it as a valuable future staple. The main barrier to adapting microalgae as a staple is its undesirable sensory profile. The repulsive odors associated with microalgae trace to volatile odorous compounds, or VOCs. Common approaches to mitigate malodor include washing, gas or vacuum stripping, and adsorption. A literature review reveals that the current deodorization field is not systemic. Results are generally subjective, reported at sensory evaluations or purchase intent scores. These outcomes reveal nothing about which VOCs are removed versus left behind. This prevents the field from advancing forward because researchers cannot identify factors that cause experimental failures and improve in future designs. Another major gap is the absence of any sequential deodorization experiments. Since different treatments are effective for different compounds, applying treatments in sequence intuitively raises VOC removal success. However, expert outreach confirms that both gaps exist. To address the identified gaps, I developed a computational model that could map VOC removal across different deodorization treatments. The model targets spirulina, a strong candidate for a future staple. Washing, activated carbon, and ozone treatments were chosen to be mapped in the model. These treatments cause little nutrient damage and are cost effective enough to be scalable commercially. The model is constructed from different chemical principles that govern the mechanism for each treatment. Necessary parameters for VOCs are found in past research or calculated through molecular models like the QSAR. The deodorization model consists of two layers. The class layer tracks groups of compounds that share similar chemical properties and covers the entire VOC profile. The individual layer is a supplementary layer, prioritizing selected compounds with particularly intense odor. Highlights of model results are presented here. For the washing and activated carbon treatments, certain experimental factors are found to be irrelevant to VOC removal after some threshold. The removal becomes chemically saturated. Ozone treatment convert parent VOCs into daughter compounds, so it is actually possible for VOC concentration to increase. Model predictions identified volatile acids and saturated aldehyde content to increase after treatment, which aligns with experiments reporting more intense odor after ozonation. Overall, the model displayed changes to the entire VOC profile across varying experimental factors. Predicted concentration data could serve as references for future experiments addressing the identified gap. Contrary to common beliefs, model predictions reveal that chemical incompatibility is not the major cause for incomplete VOC removal. Instead, focus should be shifted to kinetic or physical factors. Additionally, the model's group level outputs presents group treatment affinity, identifying which treatment is most effective for targeting each VOC group. This supports future designs of treatment sequences. The model is limited by data collection. Since research in the field is scarce, many components of the model are borrowed from air or water quality research and might not transfer cleanly to spirulina deodorization. The ability to optimize experimental conditions in any way is new to the deodorization field. Although the model does not guarantee 100% success, it does significantly improve chances of breakthroughs because it allows for design improvements for the first time. The barrier to consumer demand is objective, not preferential. The sensory profile of whole spirulina biomass is biologically repulsive to human, so effective deodorization has great potential to raise demand, attract investment, and increase production. My next steps are to further improve the model and refine it as an effective prediction tool. My ultimate goal is to develop a treatment pipeline that algo companies could follow to effectively process spirulina. Expanding production will require further marketing and scaling, but a systematic approach to deodorizing spirulina will be the first step to a more sustainable food system. Thank you for listening.